Welcome to the Renal Research Institute's Frontiers in Kidney Medicine and Biology, where we share knowledge and advances in kidney research with the world. In today's episode, we discuss the principles of aging, and we will talk about what we can learn from the animal kingdom in order to identify novel opportunities to slow down the aging process in humans. My guest today is Dr. Peter Stenvinkel, Professor of Renal Medicine at Karolinska Institutet in Stockholm, Sweden. Dr. Stenvinkel has published more than 600 articles and reviews on various aspects of inflammation, wasting and metabolism in chronic kidney disease. So hi, it's a great pleasure, Peter, that we have an opportunity to talk today about aging. And I don't know about you, but when I looked this morning into the mirror, I saw my wrinkles and I said, oh gosh, I'm really aging. But this begs the question, of course, Peter, why do we actually age?
Well, actually, Peter, humans are living longer than ever before. And in fact, newborn children in high-income countries can now expect to live more than 100 years. And the natural conclusion is that both the miracles of modern medicine and public health initiatives have helped us to live much longer than before. But maybe now we will not extend further, because I just realized reading about the literature here that life expectancy have now stopped to increase. That was confirmed in UK, and maybe also in other countries we are now seeing that the gains we have seen are slowing worldwide, and of course the COVID-19 pandemic will also contribute to this.
I'm wondering, Peter, has actually the maximum lifespan of humans changed over the millennia? We know that people got, I think the oldest around 120 years, but in the past, say 100 years, 500 years ago, say, did people also get that old? I'm not talking about the average lifespan at birth, of course, but the maximum lifespan. Has there been a change?
Not really what we know, because of course these data are very hard to scientifically prove, and the human being that we have the longest documented living, that is a French woman, Jeanne Calment, and she was born in the late 1800s, and she was living 122.5 years, which is an amazing lifespan. But we don't really know further back than Jeanne Calment if there's been a longer lifespan than she have had. So this, in essence, or biological aging, is of course the deterioration of functional characteristics, and the causes of aging are present throughout our entire lifespan, and there is no going back. So this, in essence, changes we see increase the risk of pathologies, morbidities, and mortality, and that's why we sooner or later will die. But if we could stop senescence for some magical reasons, the majority of us would reach 100 years old.
This is a very interesting outlook. I'm also wondering, Peter, do you think it's really nature or is it nurture that defines the lifespan, a combination of both? What's your thoughts around that?
Well, I would believe that it is both of them. There is a much discussion among researchers about the mechanisms that contribute to the aging process. It is widely accepted that damage to genetic material, cells and tissues that accumulate as we get older which cannot be repaired by the body, or that's the causal factors for the functional decline associated with old age. But what causes this damage on the molecular level and why it can't be repaired in old organisms, that is much less clear. There is a potential to repair in younger species, but not in old organisms, and that is one of the major research questions that we really need to solve now.
to me it was always fascinating to see that mammals age at a different rate. So small mammals usually live shorter than large animals. Is this actually known why that's the case? Interesting enough, I mean, I've also read that the number of heartbeats per lifetime in a mouse seem to be the same as in an elephant. So I'm always wondering there should be or must be some more profound underlying biological principles. What are your thoughts on that?
I find it so amazing that the lifespan, that it varies so much between animals. It can range from only a few hours in mayflies, and we have Icelandic clams with a documented age more than 500 years. And actually there are some primitive animals like sea anemones and the freshwater polyp called the hydra that does not age at all. And we also have other amazing observations, for example the Greenland shark that can live up to 400 years and reaches sexual maturity at the age of 150 years. So there are many different features that are related to age in the animal kingdom. And of course we can learn a lot from these observations. We know that the bigger animals, they live much longer. The scaling exponent for the relationship between lifespan and body mass is between 0.15 and 0.3. And as you mentioned, the number of heartbeats is also something that is related to age. We know that there is a quite strong inverse correlation between the lifespan and the heartbeat. For example, in long-living whales, there is a heartbeat of 20-30 beats per minute. It's interesting that humans fit very poorly into this regression line because we live a comparable long life, but we also have a quite high heart rate among 80 or 90 beats per minute. Other important features that is related to aging in the animal kingdom is related to loss of telomere length. It's very strongly related to aging. We know that reactive oxygen species is low in long-lived compared to short-lived animals. Long-lived animals have less endogenous antioxidants and much less mitochondrial reactive oxygen species than short-lived ones. And we also have a really recent observation that is not yet published, showing a strong relation between the number of somatic mutations in the animal kingdom and lifespan. And for us nephrologists, Peter, I think it's just a fascinating observation that phosphate levels is so strongly related to lifespan in the animal kingdom. So the higher the phosphate levels, the shorter the lifespan. And there are numerous studies showing that phosphate actually promote aging processes.
I mean, Peter, you have done most interesting research in a variety of animals. I remember your studies in hibernating bears, for example. I guess you were one of the few nephrologists who ever would crawl into a bear cave. Was this kind of research also related to aging, or did you pursue other questions on that?
Well, the original reason for my interest in hibernating bears was there was a ranger in Yosemite National Park that once told me that hibernating bears do not pee for 4 to 5 months during the hibernation period. And for a nephrologist, that is just amazing that the creature can sleep for such a long period without eating, drinking, not peeing, and waking up in the spring without losing much muscle mass, without getting osteoporosis, without signs of inflammation or vascular disease. And this was the main reason I got interested in this. And then collaborating with bear researchers and zoologists and veterinarians, I realized that there is so much to learn from ingenious solutions in the animal kingdom, and the hibernating bears is just an excellent example how a creature can avoid burden of lifestyle diseases that we see in us humans. They get extremely fat during the late autumn in preparation for hibernation, but this is an obesity that is associated with less insulin resistance, which I find just amazing. And we just recently discovered also that they have seemed to develop a natural immunization process that protect them against atherosclerosis.
So this is now really fascinating. So because this would be a sort of an anti-aging mechanism also. Okay? I don't know, because you said it hasn't been published yet, if you can say a few words about that. Otherwise, we wait for your paper on this.
Well, we have in this paper that is now published, we looked at the oxidation of cholesterol, and it seems like they have developed antibodies that decrease the risk of oxidation of cholesterol, and there is an enormous difference in this levels of these antibodies during the summer active period and the hibernating period. But of course we need to understand why they develop these amazing high levels of antibodies during the hibernation period.
Okay, yeah, I mentioned at the very beginning, when looking into the mirror, of course, everyone who ages sees wrinkles, which brings me to the question, Peter, do organs age actually at a different rate? I mean, can I tell from the wrinkles in the skin something about the age, say, of the liver or the bones or the kidneys? And is there a relationship with or some differential organ-specific aging rate?
Well, that is an extremely interesting question. And in communicating chronic risks, there is an increasing use of a metaphor that can be termed effective age, or the age of a healthy person who has the same risk profile as the individual that you would like to evaluate. And these are some discussion about real age, heart age, lung age, kidney age, and so on. And I think as a nephrologist, we actually see that patients with chronic kidney disease, they have a different in the rate of organ aging. And we are all aware of this perfect storm for accelerated vascular aging we see in our patients. And of course, this must be a number of different risk factors, including hypertension, low-grade inflammation, disrupted mineral metabolism, oxidative stress, lack of vitamin K, and somatic mutations, which is an area my research group is now more or less interested in. But I without having much evidence in the literature, I would say that organs do age at different rates, but the reason for this we don't really know yet.
Yeah, because this I think would be an important area of research to understand what prevents aging in one organ versus another. And I just try to think clinically. I clinically, and I don't know if you share that view, sometimes I get the impression that patients with congestive heart failure have an accelerated aging, or patients with chronic inflammation, chronic inflammatory diseases, even without kidney disease, which brings me actually to the question, I mean, what's the role of inflammation, I mean, in the aging process? I mean, we covered genetic reasons or indicators like shortening of telomere lengths. But could it be that chronic ongoing inflammation is the overarching theme when it comes to aging?
Yes, that is my belief. And of course, inflammation is acting together with its partner in crime, oxidative stress. They are always linked together. And there is a kind of vicious circle of inflammation, oxidative stress, that often goes together with hypoxia and mitochondrial dysfunction. They are very closely related. You will see them more or less disturbed in most burden of lifestyle diseases that we know tend to aggregate as we get older. And of course our patients with chronic kidney disease, they also often have multimorbidity, heart failure, diabetes, obesity, congestive heart failure, Alzheimer, you name it. And inflammation is, of course, the common denominator of all these burden of lifestyle diseases. We also see, and this may be an important link, that there is a depression of the expression of the transcription factor NRF2 in these diseases. And another feature which I find extremely interesting is the gut microbiota, because inflammation and burden of lifestyle diseases all seems to be associated with the disturbed microbial composition and the leakage in the gut that may contribute to inflammation. And I think here we can actually learn a lot from the animal kingdom, because animals that tend to live longer, they have developed ingenious mechanisms that counter inflammation, oxidative stress, and gut dysbiosis.
So I think that one of the animals you were particularly interested in is the naked mole rat, as one of those animals that seem to be almost resistant against inflammation. Is there actually something that we as humans can learn from certain animals, how they defy aging or decelerate the aging process? What would be kind of lessons for us as humans?
Well, there is a lot we can learn, and together with a gerontologist, Paul Shields, in Glasgow a couple of years ago, we really searched the literature to understand what characterized long-lived animals, and that is animals that live much longer than would be anticipated for their age. And here, of course, snake mole rats, they stand out because they have the size of an ordinary laboratory mice or rat, but they live up to 35 years, and they don't develop any signs of vascular aging. They are almost resistant against cancer. And there is now a lot of new pieces in the puzzle that we have learned from these snake mole rats, especially regarding their protection against inflammation. And they have a markedly increased expression of NRF2, which opposes the transcriptional upregulation of pro-inflammatory cytokine genes. They seem to have a highly efficient mitochondrial function and also superior maintenance of protein homeostasis. Because as we get older, our proteins are modulated. They are affected, for example, by carbamylation, glycation, which is you will see in diabetic patients. But the protein homeostasis is really maintained until very high age in these naked mole rats. So there's a lot we can learn from the naked mole rats.
So low caloric nutrition actually has been shown to extend lifespan, I think, in mouse models and maybe in other rodent models and maybe in other animals. Do you think that this is one of the lessons that we could learn to use low-calorie diet as a means to decelerate aging?
Yes, you are completely right that compared to control animals, a caloric restriction extend lifespan by about 50%, and that is found in a large range of different mammals, all the way up to primates. So the reduction of the metabolic rate, it has been hypothesized that caloric restriction could extend lifespan by decreasing the rate of free radical damage. So I would guess that caloric restriction also would be operative in humans, but a problem with this strategy is that caloric restriction may have adverse effects, and of course it will reduce the quality of life in humans. You will need extremely long-term studies in order to prove this, and I think it will be very hard to find humans that would accept a markedly calorie-restricted diet for many years. So therefore, I think an alternative approach to arrest aging, which I would find much more interesting, is to find and use drugs that have caloric restriction mimetic effects, which are pharmacological agents that we already know many of them, that could recapitulate the main biochemical properties of caloric restriction, which is then, of course, the global reduction of protein acetylation and the induction of autophagy.
So when one would use such kind of drugs in a trial, you need, of course, markers of aging and to show differences in the various groups. So are there currently well-established markers that would react in the short term to changes in the aging process?
Well, there are a number of surrogate markers of aging. We don't have a golden standard yet. And of course, we have surrogate markers for vascular aging that we have been used for many years, such as endothelial dysfunction and pulse wave velocity. There are a number of metabolic markers that have been used to assess biological age or aging processes. But so far, there have been really no acceptance of a golden standard. I personally think that DNA methylation markers, the epigenetic clocks, is so far the best way to study changes in aging accelerations, for example, and to find a marker of biological age. But unfortunately, these are extremely cumbersome and expensive techniques that doesn't really allow yet us to do studies with nutritional or pharmacological interventions. But my research group at Karolinska, we are currently trying to evaluate biological aging in patients with chronic kidney disease to see how it relates to risks and outcome.
Yeah, it's very interesting. So we are certainly looking forward to the results of your research. Now, Peter, I think you were certainly for me the first one who kind of would connect global warming or climate change to potential to aging processes, and it was really fascinating for me to see you making this connection. Actually, this connection might be new to many of our audience members. So may I ask you, Peter, just to enlarge a little bit on that? How is climate change in your mind related to aging?
Yes, so we are exposed to the exposome, something that affects aging processes, and there is an intermediate inflammatory phenotype, the one I described before, including persistent inflammation, oxidative stress, mitochondrial dysfunction, tissue hypoxia, that is, we know, stimulated by many of the well-known risk factors, sedentary lifestyle, nutritional factors, etc. But there is now more and more evidence, unfortunately, showing that also our external milieu will stimulate this intermediate phenotype with inflammation, and I'm particularly thinking of global warming. Air pollution, loss of biodiversity may also affect this. And we have in so many different disciplines now had reports that show that burden of lifestyle diseases that accumulate with age will be triggered by the changes in the environment that we now see. A report from Brazil a couple of months ago actually showed that there is a dramatic increase in the prevalence of chronic kidney disease that is directly related to heat waves in Brazil. We have since many years been known that agricultural workers, often young men in Central America working in extremely warm conditions, have an increased risk for chronic kidney disease. And this is something that we see in so many different fields now, and there's an emerging discipline, the planetary health, that we're trying to connect all the dots and see how these changes in our environment will affect burden of lifestyle diseases. So today I just read an really interesting study from in Nature evaluating changes in food consumption in China. And we know that there is no other country in the world where the meat consumption, the red meat consumption, has increased so much as it has done in China. And we know that meat consumption is and meat production is something that actually have a high environmental impact on the carbon dioxide footprints. And they calculated that if the Chinese would actually follow the rules or the guidelines set up in China's eating habit rules, that this would have a major impact on air pollution. So there are intricate links that we have not really been aware of, and there are studies also showing that global warming will affect the gut microbiota. We know that air pollution will affect the gut microbiota composition. So there are so many emerging threats that could affect this whole cluster of burden lifestyle diseases that we need to understand much better.
Yeah, so I mean, I'm wondering, as humans, we can shield ourselves to some extent, say, against warming by using more air condition and changing clothes, etc. I'm wondering if animals that cannot protect themselves as good against climate change and with, of course, a much shorter lifespan, if certain animals would be actually appropriate areas of research to look what climate change indeed causes in terms of physiology and pathophysiology.
Yes, it's a very good question, and there is my favorite animal now when trying to understand what we can learn from animal kingdoms are the bats. These are ingenious animals that have developed so many interesting mechanisms during evolution to protect themselves that we actually can learn a lot from now. And this is, of course, now during the pandemic, and the bats, they have a very robust interference system, and this helped them to persist virus infections. We know that bat immune defenses drive the evolution of faster transmitting viruses. We have seen many examples of this, not only COVID, but also Ebola virus. But the bats are protected against the harmful effects of their own prolific viruses, but other species, such as we humans, are not. And to understand why the bats have developed this ingenious system, we should understand that bats is actually the only mammal that we know of that have learned to fly. So they are capable of true and sustained flight, and flying comes at an expense, and that is an enormous increase in the metabolic rate. It's actually increased 15 to 16 folds during flights, and this is compared to only a twofold increase in metabolic rate in birds. So if you have an increased metabolic rate, that will promote cellular DNA damage. And in order to survive this cellular DNA damage, they need a positive selection to have an an interior milieu of low inflammation, which will lead to an extremely tolerant immune response, and then of course risk for virus infections. But at the same time, they have this extremely robust interference system. So bat seems to have found out how to protect themselves against virus infections, and this has a huge relevance for us humans, because we know that the main problems with COVID-19 is this hyperinflammatory syndrome, and in combination with an poor interferon response. So there are new studies that have shown that bats have targeted the inflammasome pathway at multiple levels, which mitigate immune-mediated tissue damage, and at the same time they have a markedly increased expression of the transcription factor NRF2. And this may also be one reason why bats live about 4 times longer than animals with a similar size. So that they are really, really interesting for us to understand how we should deal with future pandemics, which again I think is just a sign of a poor planetary health.
I mean, listening to you, Peter, I have to say it's just, we may need to rethink in a way how we approach these questions, just to look so carefully into what solutions has the mammalian kingdom found actually, instead of, for example, using animals predominantly for, say, testing drugs, but just to really take a look at animals in their natural habitat and see what lessons can be learned from them. And I think this is one of the, to me at least, one of those take-home messages from our conversation here. Now, I see that we are approaching the end of it. Peter, may I ask you just to summarize in a few sentences messages about aging that are really important to you?
Well, to me, I really found now after studying the literature here that aging is extremely complicated. We have different hallmarks that contribute to the aging processes, and we need a lot of studies here to find out which are of importance. And also, I think we have been so focused on lifespan, but what we should really address is health span. And we need to increase the health span, because this long period in patients with burden lifestyle diseases for 10, 20, 30 years when they have chronic kidney diseases, the difference between lifespan and health span is extremely troublesome, not only for the patient per se, but also for our society. It's extremely expensive to have these long periods of chronic diseases. So what we should focus on is not to increase lifespan, we should increase the health span and try to shorten this very costly period. And here I am convinced that we have so much to learn from ingenious solutions that have been developed in the animal kingdom. That they have different species have develop protective mechanisms for almost all of these burden of lifestyle diseases that we are now having so much trouble with. So instead of destroying and using nature, we should actually learn from nature, because there are, I'm sure, many different solutions out there that we just need to find out if we treat nature in a good and positive way. So I think that kind of summarized what I believe in this area.
Yeah, no, thank you, Peter. You made a strong point, a very good point that also emphasizes that maybe we have to rethink our relationship to nature, to the environment in a way, and that will allow us actually to really learn from them and from their millions of years of evolutions and from those ingenious solutions that evolution had helped mammals, other animals to actually develop. So I would want to thank you, Peter, really a lot for this conversation. It was wonderful. It was inspiring to me. And I hope also for our audience. So thank you again. And I'm very much looking forward to future results of your research and also of research of other colleagues in this area. Thank you.
Thank you. Bye-bye, Peter.
Thank you for joining the Renal Research Institute for this episode of Frontiers in Kidney Medicine and Biology. We invite you to engage with us on our social media channels and look forward to seeing you again for the next episode on Frontiers in Kidney Medicine and Biology.